So this is a complex topic because indeed these are a rare population of patients. And when we have mutations in BRAF, usually they are classified into 3 different classes according to the alterations in the protein and also the protein function according to these different mutations. So I have the class 1, which is the V600, namely the V600E, which is the one that we know the most from melanoma and other solid tumors, but we also have V600A, K, D, and in general they are sensitive to first generation BRAF inhibitors that now we also combine with the MEK inhibitors, so dabrafenib, trametinib, or encorafenib plus binimetinib. And why are they effective is because these class 1 mutations lead to an activation of the BRAF protein, and she becomes active as a monomer, so all alone, very active in the cytosol, and being active and activating all the MAP kinase pathway until the nucleus to lead to cell proliferation and survival. And it's doing that in an independent way from the RAS signaling, which is upstream. On the other hand, when we have a class 2 mutation, what happens is that these RAF proteins also become active, but they have to form dimers or couples, so they have to be together to activate the downstream pathway, and they do it also in a RAS-independent manner. But because they are a dimer, if you just give a first generation BRAF inhibitor, that won't work. Why? Because the BRAF inhibitor connects to one of the partners of the couple, let's say like this, and this leads to a conformational alteration, so a change in the structure of the second partner of the couple, and makes it resistant to the BRAF inhibitor, which means that it actually becomes active and leads to this activation of the downstream pathway. So that's what we call the RAF paradoxal inhibition. And finally, we have the class 3 mutations that what they do is actually they'll lead to a low active or an inactive kinase protein. So this BRAF couples itself to RAS in a tighter way, and so it amplifies the signals that come from the RAS activation to the downstream pathway. And that's why it doesn't work to inhibit BRAF, also because they form dimers, and so it's the same thing from the class 2.